Viruses. This is Chapter 9, Section 3. What exactly are viruses? They can be defined as acellular microscopic particles. That means that they're not made of cells and they're really small. Smaller, much smaller than even bacteria, which this little image up here illustrates. You've got, viruses that are here, the little tiny red speck, which is smaller than tobacco smoke, which is really much smaller than bacteria, and fungal spores, and plant spores, and rain droplets. So they're itty bitty. They have genomes that consist of nucleic acid, either in the form of DNA or RNA, which is ribonucleic acid. They replicate inside of host cells using the host metabolic machinery. All that means is that they use host parts rather than virus parts to kind of hijack and make new viruses. So again, they replicate inside of host cells using host metabolic machinery and host ribosomes to form a pool of components which assemble into particles called virions. And these virions will protect the viral genome and are used to transfer it to other cells. All right, so here's another picture on size. Shows parvovirus right here, which is really small, next to a nanobacteria, which is a small bacteria, next to another bacterium, next to a really large virus that they discovered infects amoebas, and to a really large bacterium found in the ocean. And then you get to average sized E. coli, which is quite a bit larger. So viruses are small. If we look at comparing viruses to cells, there's several differences. Cells have lots of, at least eukaryotic cells have lots of components, but viruses just have basically inside of them DNA or RNA inside their core, surrounded by a protein capsid. Viruses can only reproduce within a host cell. Cells can reproduce on their own. Cells are always DNA, but viruses are DNA or RNA. Viruses do not grow and develop. They're assembled, as we already said. Cells do. Viruses don't really obtain energy or use it. They might have a little bit stored that they took from a cell. Cells, of course, can do things with energy. Viruses don't really respond to their environment much, and cells do. And as far as changing over time, yeah, both of those do actually do that. And we'll talk more about that in a minute. As far as structure goes, viruses consist of a core of genetic material that's either RNA or DNA, like we just said. They are surrounded by a protein capsid, which is this sort of bluish purple guy here, and can come in different shapes. Around that is often an envelope, which can be made from host membrane. And then attached to that envelope are usually some sort of protein or carbohydrate surface marker things that help the virus invade their host. So let's see if we can guess these parts. So these little dots on the outside, these spikes, those are the envelope protein or surface proteins that they use to help sneak into cells. This little bit here, does anyone remember what that I just said that is? That is the envelope, which again can be taken from a host membrane. This inner wiggly guy here, that is your viral genome, or the DNA or RNA. Out surrounding that is the capsid. Here, capsid is fine. You don't have to know nucleocapsid. And then just outside of that, this is something you don't really need to know, is the viral tegument. All right, you don't have to know that one. All right. The infection cycle usually involves a virus coming up to a cell. And by the way, this is not drawn to scale. Okay, viruses are much smaller than this. They enter the cell either directly or they may just inject their genetic information into the cell. The genetic information gets into the cell's machinery, usually if it's a eukaryotic cell, to the nucleus, where it sneaks in with the rest of the DNA and then starts making proteins that the virus needs. And basically it hijacks the cell and starts producing all these different parts that get assembled together until you get a whole bunch of new viruses made, and then they're released to go infect new cells. And that usually causes the original host cell to die. Not very nice little guys. How do these viruses trick the cells into letting them in? It usually involves an interaction with the host surface proteins, these little cytoplasmic membrane proteins. Viruses are very good about producing a variety of different surface marker proteins that can interact as a key. They're sort of like little thieves, all right? They can pick locks. They have these structures on the outside of their capsid that allows them to trick the cell into thinking that it has the right key, and so the cell will recognize it as something that's okay to come into the cell. And then as soon as they get their genetic material into the cell, it's all over. The cell's been compromised. Viruses are classified according to the following. They're classified by their shape. Shapes include crystal, cylinders, spheres, and spacecraft, and I'll show you those in a second. They're classified according to the type of disease that they cause, to their life cycle, whether they're a lytic cycle or a lysogenic cycle, and I'll explain those in a second, or the kind of genetic material they have, whether they have DNA or RNA. So as far as viral shapes go, here are the viral shapes. And I just said you had cylinder. Well, let's see this one. Crystals is one. So which of these do you think looks like a crystal? Hopefully most of you will pick this one. Then we have cylinders, and that's this helical shape down here. That's a cylinder. Spheres, that's that guy. Influenza. Spacecraft is this guy. Most of the spacecraft are actually bacteriophages, which are a group of viruses that attack bacteria only. They don't attack eukaryotic cells. They only attack prokaryotic cells. Pretty cool. Types of viral diseases, you don't have to know these really, but if you could know one example, that would be fine. There's respiratory infection viruses. There's GI viruses, which are ones that affect your stomach and intestines. There's exanthematous infections, which it's a fun word, affect the skin. Things like measles and chickenpox, I think, are in there. Hepatic infections, which affect the liver. Neurological infections, which affect brain and spinal cord. Hemorrhagic fevers that affect the blood and connective tissue. This is where Ebola and Marburg and stuff are located, is in this group. Cutaneous or mucosal infections that affect the deep skin. And then there's multisystem diseases and nonspecific diseases. There's lytic and lysogenic processes. The lytic cycle exists basically is sort of like the general infectious cycle that we talked about before, where the virus gets into the cell. It takes over the cell's genome, it starts producing virus parts, they assembles them, and then it breaks out, killing the cell. That's the lytic cycle. The lysogenic cycle is where the genome gets incorporated into the genes of the host cell, and then it just sort of lies dormant there, and every time the host cell replicates, that viral DNA or RNA gets replicated with the host cell, and so basically the host cell is like an infected viral cell, but it's not active. It's sort of lying in wait. It's sort of dormant until at some point something might happen so that it enters the lytic cycle again and starts causing problems. As far as fighting viruses go, your immune system can be involved in a couple of ways. It can cause fevers, and fevers can prevent your body from doing certain chemical reactions, because some chemical reactions need a particular temperature range. And that means that it can slow down the production of new viruses inside of your cells. And then more specifically, your body can produce antibodies made by your white blood cells, and these can bind to the virus directly, which alerts the immune system that there's a problem, one, and that allows the immune system to target the viruses for destruction, and also prevents the viruses from actually entering new cells. So antibodies are good. As far as making vaccines go, vaccines are made from weakened or dead viruses, or possibly their protein coats alone. And it allows the body to create antibodies without fully getting sick, because basically you've got all the parts for the body to recognize without the sort of active parts that cause you to get sick. And then once you have those antibodies, if you get a real attack, your body is immediately ready to respond, and you're able to fight them off without actually getting majorly sick. And since viruses can reproduce so quickly, sometimes there's a problem with some vaccines, and that the genetic information can change enough within the virus that it can change some of the structural parts of the viruses. And if those structural parts, like the protein coat, which we use for making antibodies, gets altered, then those antibodies that used to recognize the virus no longer work. And then we start getting sick again. So this is why some vaccines are no longer effective after a period of time, and why you need to get new vaccines more often. So this is why you get flu vaccines every year, is because the viruses can kind of mutate fairly often. And just a review of how how vaccines work. Basically, you get this weakened form put into your body. The body will make the antibodies. The antibodies are then ready to attack the real virus if it happens to come into your body. There's some controversy lately about vaccines and vaccinations. Some people are having problems with it. So, if you have a lot of people that get immunized, we've ended up pretty much eradicating smallpox, which is pretty impressive because it used to be a really bad disease and problem on the planet. But if you introduce them in, then you have a situation where, like here, herd mentality. If you have enough people that are vaccinated, like here, then if somebody gets sick, there's like a shield almost around them of people who can't get sick, so that this virus can't make the jump to enough people to kind of infect a large part of the population. Whereas here, if you don't have a lot of people that are immunized, these blue guys, and somebody gets sick, then like everybody gets sick just about. Or if you only have a few people that are immunized, these yellow guys, then there's still enough people to keep infecting that still the majority of the population gets infected. But if you have enough people infected, and then there's a few people who can't get infected due to immune problems, or they're too old or whatever, you still end up protecting almost everybody from the disease. All right. And this information about, vaccines causing autism, they've, later determined that, there really isn't a connection. The original paper that was made was retracted, and there was allegations of fraud and all sorts of problems with the scientists who published that paper. And yet people are still kind of wary and worried. Whereas we really need to remember this situation over here, that, we can really protect everybody from this problem, especially those that can't get immunized, from getting a really life-threatening disease if we actually get our vaccines. But, there's still, we can talk about this more in class. So I want you to kind of think about this a little bit, because we may be discussing it in class soon. All right. I think that's all I got. Talk to you later.